By the year 2030, there will be more than 8 billion people in the world, and this will mean an increase in the demand for food. Science and business form a perfect combination to meet the challenge presented by this scenario. Agro-Agriculture and Nutritional Genómica Centro, Araucanía. A large part of the agricultural activity of the 9th region of Chile is concentrated in traditional crops intended to satisfy the domestic market with little added value and virtually no production for export. Meanwhile, salmon farming and the food industry in general, both in Chile and worldwide, urgently need to replace part of their raw materials with alternative ingredients, particularly of vegetable origin, as a consequence of restrictions in the supply of fish meal and fish oil. This situation has given rise to the Regional Consortium of Araucanía, an institution which has received investments in excess of $4 million, contributed by the World Bank, the National Commission for Scientific and Technological Research, CONICIT, the Regional Government of Araucanía, the National Institute of Agricultural Research, India Carrillanca, and the La Frontera and Catholic Universities of Temuco. Furthermore, a number of industries are involved in this initiative, both from the salmon farming sector and the regional companies who wish to back this innovative project. The research activities of the consortium begin in the Plant Biotechnology Unit of India Carrillanca, which is working to gain understanding and knowledge of the function of the thousands of genes which exist in a plant, and so improve its nutritional quality using genome analysis and genetic engineering, functional genomics and proteomics, and finally technological seed quality.
Through genome analysis, we can identify and map the genes and genomic regions related to nutritional and anti-nutritional quality in each of a plant's chromosomes. With this information, it is possible to select plants directly at the DNA level, thus ensuring that the plant has the necessary information to generate a high-value product for the agroalimentary chain in a short space of time. Furthermore, with genetic engineering, it is possible to generate genetic variability which is not present naturally in the plant, and so obtain better quality products. For example, a type of fatty acid, sulfated amino acid, a protein, type of starch. A plant contains between 15,000 and 30,000 genes. Functional genomic enables us to identify which ones are responsible for the nutritional quality of the seeds. This implies isolating genes, for example using expression libraries, and identifying them by obtaining their nucleotide sequence and bioinformational analysis. It is also possible to monitor the expression of these genes in the various tissues of the plant in order to learn the moment and location of their function. This information can be used to select plants with desirable attributes. Once we have these genes, we can manipulate them and integrate them into the plants by genetic transformation and so improve their quality. In proteomics, we try to identify proteins of interest from among all those which are present in the seed. This is done by isolating the protein, obtaining its sequence, modeling its structure, and relating this to its biological function. This knowledge will enable us to recognize fractions or subfractions of proteins with potential uses in food, nutraceutical, pharmaceutical, and other industries.
Since it is necessary to make nutritional quality compatible with productivity, environmental friendliness, and sustainability, many specific genes are acquired in every plant. This is why combined application with bioinformation processing enables us to decipher the routes which lead to the final product.
Nosotros trabajamos con secuencias de letras que nos llegan desde la unidad de biotecnología de plantas.
We work with sequences of letters which are sent to us by the Plant Biotechnology Unit. These sequences of letters identify a particular gene or protein, and we compare it with millions and millions of other chains which have already been identified and studied, and where the characteristics which make this particular gene are already known. If we are able to match the chain which is sent to us with one which is stored elsewhere, we can say exactly what its function is. With this information, we can also reconstruct the metabolic pathway. In other words, what part is played by the gene or protein in the construction or fabrication of foods and the generation of energy. and in this way we can tell how efficient this particular protein or gene is. All this information is fed back to the plant biotechnology unit so as to go on improving the quality of the seeds produced with every hybridization.
Once Karyanka obtains the product with the genetic information which has been found, this is evaluated by technological seed quality to look for new value chains starting from traditional crops like cereals.
An example of this is the development of starches with better industrial quality for the pelletizing of fish feeds and animal feeds in general. And of course, basic necessities such as biocombustibles and mass consumption plastics like ethanol, biodegradable polymers and edible films.
Another unit of this consortium is the Bioprocesses unit of La Frontera University in Temuco, which does important work evaluating and elaborating products derived from vegetable species.
In this unit, we apply bioprocessing technologies to improving productivity, putting more added value into raw materials of vegetable origin, and creating new products for both animal and human foodstuffs. For example, separation technologies and fermentation processes enable us to obtain high-value proteic fractions, amino acids, fatty acids, and polysaccharides for the use in food and pharmacological industries. This unit also plays a crucial role in the technological analysis of vegetable raw materials, which are used for the fabrication of pellets, biodegradable polymers, and edible films.
The final step in this research chain is in the hands of the Aquaculture Nutrition Unit of the Catholic University of Temuco, responsible for carrying out tests on aquatic species in order to find out how effective the new vegetable-based foodstuff is in the aquaculture industry.
Our responsibility is all aspects of the evaluation from the physiological and nutritional point of view, and also its application to reproduction and nutritional pathologies. That is, evaluating all aspects of the harmlessness of the raw materials which are used directly with the fish, in order to generate a product, in this case a fish, which is finally harmless for human consumption. We are also trying to produce food which generates the lowest possible level of phosphorus or nitrogen, which are the principal contaminating agents in the environment.
The successful development of foodstuffs of vegetable origin will make it possible to replace 50% of the fish meal and up to 80% of animal oils. This will contribute to the continued growth of the salmon industry and its expansion into new overseas markets. Furthermore, it will help to generate a 7-fold increase in the production of wheat, lupin and oil-rich crops in the Araucanía region, thus developing the southernmost scientific and technological nucleus in the world.
This initiative is a tremendous contribution to Chilean and especially regional agriculture. Here in this region, we very often have problems in competing with certain crops and because of certain situations which reduce profitability. The thing about this initiative is that it brings us new alternatives to choose from.
The important thing is that we are advancing in two areas. The government took the decision to increase investment in science and technology throughout the country. They also took the decision to do it in a less centralized way, giving more opportunity to the development of the regions.
If we want to define the CGNA, we have to talk about synergy, because united, we can produce more and better results than we can by working independently. In this way, we will produce more and better science and technology in the region. In the developed world, the importance of science for the development of a country is fully understood. This is why this scientific and technological platform explores science in depth, and each researcher of the consortium in this initiative is giving his or her best effort to convert the seed which we can produce here into a grain of knowledge for the country and for the world.
Agro-Aquaculture Nutritional Genomic Centre, Araucanía.